then we will have more and more people joining. So hello everyone, and welcome to this fourth session of our live talk series on NAMS. As Europe accelerates its transition towards non-animal methods, we may wonder how scientific breakthrough can translate into real-world adoption. And this session aims to explore how cutting-edge innovation, regulatory frameworks, and institutional dynamics must align to scale human relevant research. I am Lila Courteau, Scientific Manager at ProAnima Scientific Committee, which organizes this live talk series. Emeline is here as well. Emeline Goujon is here to help you with any housekeepings, any question you may have. Do feel free to type in the chat. The live talk is a new series of monthly events as part of the NAMS networks on LinkedIn. The aim is to gather various stakeholders around the NAMS, researchers from academia, industry, policymakers and regulators, as well as other actors, for example, NGOs, institutions or universities. The concept is really simple. For each session, we have one specific topic, one facilitator and a guest, a special guest. These two people are going to introduce themselves and the topic of the session, and then we will open the floor to the discussion. During the session, we invite participants to actively listen and participate to make the best out of this 1 hour talk. The event is going to be recorded and there will be a replay. If you feel comfortable, we invite you to raise your hand and open your mic and camera for the Q&A. And be conscious of the time to give everyone the chance to participate. If you prefer, you can ask your question on the chat, of course. We will also be using the Slido app to include everyone in the discussion. So you can join it either by scanning the QR code or by clicking on the link in the chat. And now that we are all set, I am very much honored and delighted to welcome our special guest, Hans Clevers. Hans Clevers is a professor of molecular genetics at Utrecht University and a leading figure in stem cell biology. He identified key effectors of the WNT signaling pathway and discovered LGR5 as a marker of adult stem cells, advancing understanding of intestinal biology and also cancer. He later pioneered organoid technologies, transforming biomedical research. He trained at Utrecht University and has held major leadership roles, including directing the Ubrecht Institute and serving as president of the Royal Netherlands Academy of Arts and Science. He also led pharma research and early development at Roche, where he helped establishing the Institute of Human Biology in Basel. He continues to oversee research groups at the Utrecht Institute and the Princess Maxima Center. Hans has received numerous international honors, including Breakthrough Prize in Life Science, and he is a member of several prestigious academies worldwide. So welcome, Hans, and thank you so much for joining us today.
Yeah, good afternoon.
The session is facilitated by Professor Daniela Salvatori. She's a professor at Utrecht University in comparative anatomy and physiology and head of its anatomy and physiology division. She shared the transition to animal-free innovation, TPI, Utrecht initiative, and is director of the Ombion Center for Animal-Free Biomedical Translation. Her work spans stem cell safety and disease modeling alongside the development of advancing training tools such as 3D, virtual reality, and holographic models through the Center of Excellence in Plastination and Virtual Reality. She's actively advancing the transition toward human relevance animal-free approaches in biomedical research and education. Welcome, Daniela. Thank you for being here today and being our facilitator. I will now give you the floor and stop sharing my screen so you can share your perspective and introduce the topic.
Good afternoon, everybody. Just a second, I'm gonna share the screen. Yes. I have a few slides to talk about Ombion, the Center for Animal-Free Biomedical Translation that has just born. And I would like to share with you the ambition of the center. And today I would like to focus on the validation, on the acceptance of new approach methodologies, and how which kind of strategy we could put together for lifting NAMs from development to effective use. The National Growth Fund is a governmental initiative and invested 125 million into the transition to animal-free innovation. This is a special initiative that brings together academia, but also private companies. So when we wrote our grant, already we focused on content to be developed together with companies. And our budget is already of 245 million, including the companies that would like to work with us. And we have already in the first year signed all the consortium agreement for working with the companies. So while being a academic initiative, Ombion has, for its nature, the ambition to create economic growth in the country and beyond. So it is a public-private partnership, and we have given to Ombion a special being, is a foundation, also independent for the founding partners. And there are a lot of topics that we are developing within Ombion, but I would like to focus on how to bring further the many models and systems that we are at the moment developing. Important to know that this project lasts for 10 years, and it is not only a virtual project. We have invested in a facility in Utrecht, and we would like to welcome all of you to come to visit us and talk with us. So our mission is bridging this outbursting development of models with the need of personalized medicine and more effective drug development. And building this bridge, we know that is difficult because it is from many years that we are talking about validation, qualification, and regulatory acceptance. And if we look at what has been done in the cosmetic and in the tox field, we know that this takes quite a bit of time. We know that human-based models are constantly developed, but what about the implementation? So the implementation is at the moment the main worry and the main, let's say, focus that also the current documents published by EU, I think about the Biotech Act, I think about the recently starting ERA agenda, European Research Agenda. How do we transition from this development to effective use? And there are some basic questions. How do this model compare to human biology? How can we make sure that it can be used in a reliable way? And I have taken this picture from the Biotech Act. This is also an economic story. If we don't give the right direction to NAMS development and uptake, also our economic sector linked to NAMS will stay behind. So EU is very good in strong scientific publication and base, but then when we look at investment in the biotech sector, we stay very much behind. And I see a lot of academic partners that have already joined the afternoon. When we make a new NAM, a new model, there are a lot of steps from developing the model to adopting the model. And starting from the context of use, the part related to the standardization, the pre-qualification of validation, and then the formal qualification. Still, this road remains very difficult because the knowledge is scattered, is not unified, and the knowledge also needs to be also tailored for the specific case. And when we talk and we start developing models, the key question is, what is the use of my model? And how can we measure the endpoint and justify that it is fit for purpose? And so unify the biology with the technology. How can we make our models reliable? And how can I validate if my model is relevant? And what do I need to do before going towards the formal qualification? We are in contact with the ECVAM, the European Center for Validation, that is driving the validation and qualification study, and they have the problems that the models, they never arrive well prepared for the formal qualification. And then we have also other questions that perhaps this afternoon we can discuss together. We need to be able to make this model to upscale this model. We need to make them user-friendly and cost-effective. So it is a quite complex picture that we need to deal with. Now, what we would like to use our center and let's say the energy, the money, the people, the knowledge, the know-how that it has been already put together in our center is really to bridge the development of NAMs together with the favoring the commercial use of NAMs. So our center in Utrecht will really have the facility focusing on bridging the development and the implementation. And just to give an idea about, and here perhaps collect your feedback about how we think about building this system. We work already with the REVM. We are closely following the OECD new guidelines that will be soon released. We are following the GV guidelines. We are connecting with the regulatory authorities, and we are also learning by doing, which we would like to have some clear showcases also for talking with the community, because otherwise this case stays very much in the niche. And at the same time, we have disease models that we have chosen, but that we will expand, and we are really progressing in making tools for understanding the state of development and standardization of these NAMs. We are including directly the end users. I have just mentioned some of them, and we would like to have a clear space for valorization of these models. And the automation lab, it will be one of the biggest automation lab in the country. We have already presenting it in a summer school that will start in July, and we need to start really talking about scalability and which models are more prone to be scalable and automated in a short time. And yeah, so we would like really to have this standardization and qualification toolkit to share and develop with developers and end users. Yeah, also we need to measure what we are doing. We need to be critical. And this is why this is a slide prepared by one of our postdocs. We need to be really critical about how to measure the technological readiness, the commercial readiness, when and how to prepare the regulatory readiness while thinking at the societal readiness. And with societal readiness, I mean also, for example, how to make a system in which we can have easy access and sharing of human material and human data. So very relevant and difficult topics. So this is just to say, this is the space where Ombion is. We are working with a lot of public and private partners, and we would really also reflect on how to make easier this public-private partnership while in close dialogue with the regulatory agencies. And yes, we are looking forward to connect with you, to hear from you, and to share our facility with you. Thank you.
Thank you so much, Daniela, and thank you for giving us a better idea of what you're doing at Ombion. It's really such an inspiring work for all Europe, and I think it's also very complementary with what Hans will present, basically. And I guess I don't need to tell more, and I will give the floor to Hans now. Thank you so much.
Yeah, thanks very much, Lilas, and welcome to everybody on the screen. I guess I'll need about 10 minutes. I'll give a bit of a background on how my lab developed one of the two organoid technologies, and I guess organoids are now well positioned to play a major role in what's called NAMs. I think you call them non-animal methodologies, and the EU now says new approach methodology, so a little bit broader than only non-animal. But I'll get back to that at the end of my short presentation. So this all started in our lab with studying the gut. You see here the small intestine of a mammal. These protrusions are called villi. They extend the surface of the gut, allowing you to efficiently take up nutrients. And then there's stem cells that sit between these. At the base of these villi, you see these little black holes. The holes are called the crypts of Lieberkühn and had been known for about half a century or even longer that actually probably the most active stem cells of the mammalian body, mouse or human, sit somewhere in this crypt. And what we discovered, I'll show you in the next few slides, is that these multicolored cells at the base of the crypt essentially are these stem cells. This is a different view, a real microscopic image. So you see villi, you see in blue the enterocytes that take up nutrients. In red, you see the goblet cells that make mucus. But there's at least 10 other cell types, including the cells that make GLP-1, the weight loss hormone that's now becoming very popular. Another view on a cross section, you see villi and pointing up, and then here and there you see these villi, the crypts that stick into the wall of the gut. And Nick Barker in the lab, and I won't take you into any detail here, discovered or rediscovered the cell type at the very base of the crypt. This is actually in a mouse where these cells, he made light. You see them in green, so every little stripe is essentially one of the what we now call the L5 stem cells. And they drive this incredibly fast self-renewal process physiologically, so you don't need to damage the organ. Every 5 days in the mouse, probably every 7 days in a human being, what happens is that cells are born at the base of the crypt from these stem cells that I just showed. They rapidly proliferate for about 2 days while they move up. They then become one of about 12 different cell types. They keep on moving up, and 5 days after they're born, they die at the tips of the villi. So it was the most actively dividing stem cell in the body, and we thought we should be able to culture these stem cells. Toshi Sato took this on, and we, from a number of random experiments, we knew the growth factor conditions at the base of the crypt. High wind, so we added a wind amplifier, our spondin, epithelial growth factor. We knew we had to block BMP. We do this in Matrigel, probably very well to all of you on the screen who do organoid experiments. I get back to that to the end. There's lots of problems inherent with Matrigel. So Toshi, much to his surprise, didn't get just a lump of stem cells when he started from one stem cell on day 0, but he got these structures that we now realize are like mini copies of the gut. And he originally called them mini guts, they're now called organoids. And you can actually see, so they're closed spaces that don't really look like a gut, but when you call them close together, they fuse, and you can actually make long tubes that really start resembling the real gut. And not saying too much, but these things never stop growing. However, they stay normal. When we transplant them, they make normal tissue. They never make cancers. They're also not exhausted. They have all the cell types of the tissue that we are trying to culture here. So all of the 12 different cell types of the gut epithelium. Telomeres don't get short. So there were many, many surprises, but we actually built on this. This is a liver organoid. So once we figured out how to grow this most active stem cell of the human body, we then realized we can use the same principles to grow other parts of the body. Here's liver, here's a number of examples. This is in the middle liver. Top left is airway from the lungs, and these green cells have the cilia, the beating cilia that clean mucus and debris out of the lungs. Top right, I believe, is an RSV infection in light green. Bottom right is a corona infection in a gut organoid. The virus is painted in white. And bottom left, I believe, is a cervical carcinoma. So in essence, to summarize what we can do with this technology is we can grow any epithelium of the human body. We cannot grow any other part. So no muscle, no tendon, no fat, no brain. That can be done from iPS cells. So this is the other technology that I'm not mentioning here. The advantage of this is that actually you can have your specified cells right away from an individual. So they really represent the individual. You have your organoid in a week. You can then grow it as long as you want. Compared to cell lines, essentially they have all the cell types that differentiate the cell types of an organ. And if you think about why only epithelia, all functional parts of organs, so the organ-specific functions are basically always executed by epithelial cells in the lung, in the liver, in the pancreas. All other cell types, stroma, immune cells, nerves, blood vessels, support the organ, but they don't perform the organ-specific function. So if you're interested in organ-specific functions, epithelia are good enough. Many of the infections actually happen, I'll show that, happen through epithelia. Also in cancer, carcinomas, by definition, they're epithelial, account for about 90% of all human cancers. So with this technology, we don't capture all tissues. Brain and retina in particular, we cannot grow. Muscle, we cannot grow. But we capture many of the relevant tissues for drug development and for personalized medicine. Personalized medicine, so it looks obvious that when you can grow the normal cells, you can also grow the cancer cells. You see that here. So this is an individual with a colon cancer. Side by side, we can grow the healthy cells from this organ and the disease cells, the cancer cells. So that's also novel to this technology. The healthy cells could never be cultured. And we can directly compare for this individual, for instance, DNA sequencing, but we could do that directly on the tissue. You wouldn't have to grow organoids. But more importantly, we have the bad guys alive in the lab. So we can now actually do all sorts of experiments with them, maybe develop drugs, but also perform personalized medicine. Ombion has done a lot of personalized medicine using organoids for cystic fibrosis, but the same thing for cancer. You can just grow them as if they're bacteria, and rather than adding antibiotics, we add cancer drugs, and you can then score sensitivity or resistance to these drugs. This is it's probably now 60, 70 papers that describe that this is a highly predictive approach. However, it's very slow, it's very expensive. You need highly trained personnel. So this needs a lot of engineering, we think, to create instruments that actually automate this and do this at large scale in a miniaturized version. As you can see, we can grow a whole slew of carcinomas, and there are many more to come. And for instance, for colon cancer, take rate is about 100%, close to 100%. So it's a robust assay. Here you see normal tissue and tumor tissue from 2 patients, top and bottom. A few more things. So you can actually add immune cells. These are CAR T cells. This is a breast cancer organoid in yellow. And you see here is a discovery from Anne Rios, one of our collaborators in Utrecht. So she found out that a small subset of CAR T cells is exceedingly good in killing cancer cells. And you see one of them here. She calls them the serial killers. Every time that a cell turns red, it is killed. So 5 hours in, it probably already killed 10 cells. Now it kills another 5 or so in, and then it disappears into the sunset. So you can essentially combine organoids in their simplest form, which is really the form I like most. But you can combine it with defined immune cells, defined infectious agents, etc., etc. And I think one example of an infectious agent, because they are human, you can actually address human diseases. There are now many examples. One here is an infectious agent called Cryptosporidium. Could not be cultured in the lab. It is a chronic intestinal infection in cattle and sheep, but also in humans in lower income countries. Quite common. So you can order the oocysts that you see here. They are like the eggs of this parasite. It's a malaria-related parasite with a very complex life cycle, asexual and sexual, that you'll see in the movie. So you buy them from a farm in Texas that infects cattle and then harvests the oocysts from the stool of this cattle. We inject it into a human intestinal organoid. It goes through the entire life cycle. You see that here, so type 1 neurons. Nobody knew what these things actually infect, but they infect mature enterocytes. They do not occur in cell lines. Also, they don't infect mouse enterocytes, for instance, so there's no good lab animal model for this parasite. It goes through a number of desamplification rounds that you see here, and eventually, so after this, it will now go into a sexual cycle, so it produces sperm-like cell and then oocyte-like cell, you see that here. Fertilizes the oocyte that's happening here, and then basically completes its life cycle and is ready then to infect a human being. Yeah. So you can do that. And then finally, this is, I think, my last slide. Organoid technology is complex. I already said this. It's slow. The average undergrad student doesn't know how to do this. It's also not animal-free, because even if all the growth factors are recombinant or small molecules, a matrigel is produced by growing a cancer cell line in the abdomens of mice, and then it's purified from whatever comes out. So it is mostly extracellular matrix, collagen, laminin, but it has hundreds of other contaminating proteins. It involves animals, big batch-to-batch differences, very expensive. And we've been looking, like many other labs, for an alternative, and we found this molecule. You see it here, Invacin. This is my only slide I have in this. It actually does exactly what laminin does, so it activates integrins on epithelial cells. This keeps them alive, otherwise they will die if the integrins are not triggered. It also allows them to polarize. It also allows us now to grow epithelia in 2D. And you think organoids are 3D. Well, epithelia are 2D structures. They are sheets, if you think about it. They're sheets that are basically shaped in space, but they're essentially 2D structures, and their functions are also 2D. They're always apical to basal or basal to apical. Secretion, uptake, hormone secretion, mucus secretion, transepithelial resistance, etc., etc. So we're now exploring Invacin coated on transvals. It fully replaces matrigel. So far, every organoid that we've tried, we can grow on this protein Invacin. It comes from Yersinia. You make it in E. coli. It's a small protein, 20 kilodalton. You coat it on plastic, very easy. And now you can grow organoids as if they are cell lines. But you do have all the different cell types there. And they're normal to N. And even after 12, 14 passages, they're normal. When you transplant them, they make normal tissue. So we think that this would be one way to what Daniela just showed, that to standardize, to scale, to automate, etc., etc. This makes life a lot simpler. It would work fantastically well for the epithelial organoids. It would not work for brain organoids, for instance, because brain, by definition, is a tissue that relies entirely on its 3D connections. But for colon cancer, for many infectious diseases, for genetic diseases, that's what we think, and for pharmacology, very, very important in drug development. This will probably help. And maybe one last sentence. It was mentioned that I had at R&D at Roche for 4 years in Basel. I just returned in Utrecht. That's where I saw how industry struggles with these complex organoid cultures, where every lab publishes, even my own lab, every time we publish it on gut organoids, we always improve the method. So there's not a single standardized protocol. We live by variation and by innovation. We don't live by standardization in academia. And I think Ombion just mentioned plays a really good role in trying to bridge those two worlds, which are very different. And with that, I give the floor back to Lilas, I guess, or to Daniela.
Thank you,
And
Hans.
I'll stop share.
That was amazing. Really nice videos and very, very interesting topic. And thank you so much for sharing in such a clear and quick way you work. And I think it's really a good overview of the challenge of science and trying to find a nice compromise between complexity and simplicity, because we need also simplicity to ensure that we can scale up results and also bring the science to patient directly in a more direct way. Anyways, uh, and so now I just have one question to the audience, and then we can move to the discussion. So if you can, people, if you can join the Slido, we have just a really quick and easy question for you. But I think it's always good to have an idea of what people think. Our question is, so do you think that Europe can be a leader in the global transition toward human relevant research for both biomedical research and toxicology? We are really curious to know your opinion. And of course, Daniela and Hans, if you want to answer this question, either by just opening your mic, just feel free to do it.
Yeah, perhaps I can start, Laila. I think that is a very complex question, and at the moment, this has proven very difficult in Europe. And I think, but I would like to hear more from Hans, it is not only a matter of investment, it is also a matter of having a common vision. I mentioned the USA NIH initiative. They have invested much more in the standardization and validation part. At the same time, while I am a bit negative, I see that Europe is starting a lot of initiative about this. The European Research Agenda as NAMS as a priority. I think our government, and Hans was just at the ministry, perhaps he can tell us more, is also talking about validation. But what should we really do, and which approach should we take? Perhaps, Hans, you can help me with this question.
Yeah, so there are many aspects of this. There are many things to say. So first of all, organoid technology largely comes from Europe. We have really good bioengineers. So our challenge is essentially that we find it very hard, even in Holland, we have lots of initiatives, but these are initiatives where everybody ends up with one PhD student. And there's no coherent program. I was actually in China last week, where organoid technologies is fully embraced. There are dozens and dozens of new organoid centers that essentially were built up. Universities, Chinese companies do this. And in Beijing, the Chinese Academy of Sciences, who runs about 100 Max Planck-like institutes, they're building a 2,000 FTE organoid facility. Started last year. They're halfway now. They hope to be at full strength a year from now. So even though the investments that you showed in your slide, which I found very interesting in China, are still lower than they are in the US, they're already a lot higher than they are in Europe. And also both in the US and in China, dedicated large investments are possible. And that is, I think, the big challenge that the EU always has to take, take into account all of the different countries. Within a country, you have to take into account all the different universities that everybody gets equally served. I think that is our biggest problem. And then I think the emphasis on regulation rather than on content also doesn't help us a lot. I mean, I'm an old molecular biologist. I remember when the first plasmids came about and things like that. We were very afraid about recombinant DNA technology, introduced lots of measurements. As far as I know, we have not a single person fallen sick from a plasmid in the past 50 years around the globe, yet we have not basically scaled back on the regulations. And that is something that I think we have to really address. Also, how do we transfer knowledge in our fantastic academic world, which is at par, as you showed, with China and the US? How do we translate it in at least a transparent fashion into the commercial world where drugs are developed or where diagnostics are developed, which I don't think academia should try to do. I've seen this again at my company in Switzerland. It's extremely hard. It requires disciplines and coordination over 12 years to develop a drug. This is not feasible in academia. Also, it's not the role of academia. But anyway, maybe I should stop because otherwise I'll fill the whole hour with talking.
I agree with your answer. We should not do in academia. At the same time, we have the challenge to link strongly academia and somehow I say this strongly to a bit regulate academia in the development of these NAMs, because now you see this wide development and there are so many groups that are developing almost the same models and in many cases with not a clear focus on the context of use or the fit for purpose. May I ask you, and then I ask Laila also to stop me because I see a question in the chat. May I ask you if you would think about priorities in infrastructure that we should have in, for example, in our country? What should we have? Do you think that the plans, for example, that we are having at Ombion will be enough, or should we have a different concept from the one that we are pursuing?
Yeah, so a quick answer. So not because you're on the screen, but I think Ombion is really one of the few good examples of how this was sort of an acute investment, the Groeifonds after Corona. And you guys actually build a facility that is aiming to bridge, what academics do and what industry does. And I've lived in both worlds, and I fully realize that actually as an academic, optimization, standardization isn't in your business model. You need new papers, you need papers in high-level journals, and to take 2 or 3 years to standardize and to validate an essay isn't going to get you there. So actually, if you pursue that, you'll soon have an empty lab. And so I think the fact that the Dutch government make money available and other countries in the EU do this has helped. But you guys have actually now shaped a coordinated effort between multiple academic groups to set up this environment where you can validate. And so it's a good example. I think we should advertise this example. But I actually, and there's one last thought. We just submitted, there was a call from the EU on consortia, private, public, for NAM development. I'm sure there are more than one. We actually participated in one. We worked very hard, there's a lot of work. And then we heard that actually there is a 3% success rate given the number of people. So it's become extremely popular. But as you say, I don't think there's much coordination in who's developing what, and probably many people run in the exact same direction. And many other directions remain untouched. So more coordination would help.
Thank you, Hans. Laila, I follow your orders here.
No, please
so
do feel free to discuss with Hans because now it's your time to discuss together, and we will take the time in maybe 10 minutes, 15 minutes to get the question from participants. So yeah, do feel free to ask your question now.
Yeah, this is another point because the EU grants in the last, I think, years, they really have put a lot of emphasis on NAMS. At the same time, you see that academia is even in the course for validation, they are the driving force. And every time I see this grant, I think, yeah, we are doing it from the wrong angle. Perhaps we should think about giving more space to companies, that these private parties' needs would be more prominent. But then all the legal rules about public-private partnership are very difficult in Europe. What could we do, Hans? What do you think?
Yeah, I think you could probably simplify those regulations.
Mm.
So I was in a large pharma company that's very different from biotech. So I guess biotech in part fulfills this role because biotech can actually take, founders often come from academia. The ideas often come from academia. I must say, I know of almost no drug where the target of the drug was discovered in industry. Almost all the initial mechanisms, targets, etc., come from academia. It is when you're a scientist in a company, there is also not so much incentive to start exploring, these wild organoid ideas. If you know that the regulatory authorities that are on the other end of the project, that they probably also don't know what organoids are. So there's also, I think, a lot of education is ongoing currently. The big CROs that support drug development, like I thought in one of the slides, Charles River, you name them. They also don't really know what organoids are. They see that their business models are under threat if they only sell experimental animals. The numbers of experimental animals will undoubtedly go down strongly. I see it in my own institute. We developed a lot of the technology originally using animals, and now we sort of almost entirely abandoned our mice and are using entirely human stuff. But if you're in a pharma company, you know your assays that you're the expert in the company. You're not so attracted to some new wild approach that isn't really validated internally.
Yes.
And isn't seen by the regulators. So of course Europe was always the stronger force in trying to reduce animal use. It's a bit of a surprise that the US seems to be overtaking Europe
Yes.
nowadays. And there's different reasons not to use animals. So of course the ethics of using, but that's quite subjective. Some people think that if you eat animals, you can also do experiments with animals. But there's another scientific argument that many of the diseases don't recapitulate, are not recapitulated in animals. Pharmacology, the handling of chemicals by the liver and other organs differs from species to species. So if you can build the pharmacology in an in vitro human model, theoretically you're much better than if you do this in an animal model. So there's all sorts of reason to do this. Still, drug development and also diagnostics, because diagnostics is probably even later in following this, is very conservative and for good reasons. They want to be sure that what is out there is better than what they're already using. So this is going to be a trajectory where I think academia can play a role in the education of the other players in this field.
Yeah, I really like this last sentence that academia could play a role in the education of this field. We have also in the last years a lot of EU grants about education and training, but also there all the initiatives, they stay so much scattered, and somehow it looks like that we cannot collect the harvest, the fruit of all what is happening. And the same in toxicology, there were a lot of beautiful grants, knowing also the researchers, I think that they could really help also in this education. So I wonder how we could really not only doing the mapping of what has happened in the last year, but to bring forward the good things and the good tools that have been developed. Education stays also a very difficult, scattered field, because to influence education within faculties is very complex. But using education for professional, we have a lot of freedom. So also they're making good programs to link with academia. Would be very nice. And answer, how would you do this? How would you propose a project for this education for industry coming from academia?
Again, a few thoughts. So for industry, particularly for large pharma, it's very important that they can trust that the regulatory authorities will look at these NAMs in the same way that we hope that they will do. And this will be a bit slow. But so if we really want to enhance the use of NAMs, this should probably be done in collaboration. So that's where the EU can help in collaboration with mid-sized to large pharma companies, because they are the ones that FDA and EMA talk to. They don't really talk to academics.
No.
And also we can talk to companies forever, but if they feel that the regulatory authorities are not ready yet for this, nothing much will happen. So that's one thing. So something that we, in the context of a grant proposal that I just mentioned, that we came up with is so we have more or less an agreement with one of the I can't really name the journal yet, but one of the protocol journals that's very visible, to set up a collection of standardized organoid-based assays. Because nowadays, if you, from my own lab, if you read 10 gut organoid papers, all protocols will be different between the 10 papers. So there's nothing standardized in what we do. But so the idea would be that we set up probably an independent editorial committee of this collection of well-known organoid researchers, but also pharmacologists from industry, drug developers, to basically ensure, and this would be a living collection. So if improvements are being made, it'll be actually visible in this collection, and this could be a reference set of protocols that industry can use and that regulatory authorities can look at. And if they know this is protocol number 27 from this collection, has been used about 15 times now in IND applications and is validated in this and that way, I think that's really what's missing at the moment. It's very young technology, and essentially it is all over the place. Yeah.
Is a wonderful idea, Hans, and I think also the open access of this, it has a lot of wonderful points. And which parameter will you consider for the standardization? Have already a clear framework to adopt for saying, yes, this is good, this is bad?
I think what we would do is basically find alternatives for well-established tests. Like the Ames test is a very old test where bacteria are exposed to potential mutagens. Very little predictive value of what happened with the same mutagen in human cells, because we have totally different uptake mechanisms, DNA repair mechanisms, et cetera. But basically just directly, so standardize the protocol and directly compare it to the main assay that is currently being used in filings with EMA or FDA. That would be and then you have to show that it is at least as good and hopefully better than in predicting the understand. But of course, for every assay, it is going to be subjective. It's going to be different, and you need to bring experts in, like what you would do for peer reviewing.
And Hans, will in this line of thinking, you could have also situation in which the NAMs will not fully replace the in vivo test, but combination of NAMs could be used. So I think that it would be really nice to have not only to have this repository, but also to show this example and to reason on this example. I think it would be such a great help for the field. We should do it, aside from having the grant or not, I think.
Fully agreed.
Yeah. Okay, Laila, would you like to continue?
Thank you so much, and thank you. That's really interesting, and I would love to hear like an hour more of your discussion. But unfortunately, it's already 2:20, so now we can give the floor to participants, if they have questions. So we already have one for Marlene. I don't know if you want to open your mic and ask your question, or I can read it.
Yeah, that's fine. Thank you. Thank you, first of all, for this great session. I have a question because indeed, Hans, you said that organoids are slow and challenging to establish in culture. And of course, automation will improve the scalability and the ease of culturing, but not the establishment timelines. So I was wondering, from a precision medicine point of view, what is your view on drug testing on shortly cultured freshly dissociated patient material for rapid treatment stratification?
Well, again, a few thoughts. So first of all, I do not believe. The company I work with is one of the largest diagnostics company in the world as well. I discussed with their experts, the use of organoid-based, like particularly for cancer, organoid-based diagnostics, or personalized medicine, precision medicine, as you call it. That is very far away. They say basically the flow that the hospitals use for diagnostics is liquids, serum, urine, sputum. It is fixed tissue, scans, etc., etc. But live cells really do not work very well. Live human cells, bacteria works, but that's a brief culture. So for organoids to ever enter large-scale precision medicine type approaches, this will clearly need an instrument that is fast and can be an explant, as you say. You actually can derive screenable organoids in a week from a cancer, and that would be good enough for the treatment, but it's still a bit slower than antibiotic resistance testing in bacteria, but it's getting close. But I think the biggest challenge there is that basically, if say somebody takes a biopsy in a hospital, there is no workflow that actually takes that biopsy in a live form into a diagnostics lab. The pathologist throws it in formalin, and then they start thinking, and then it's too late. And they don't freeze it, etc., or they don't cool it, etc., etc. So my sense is that organoids will probably first be used in the drug development, and then the timelines don't really. You can just grow a large bulk of organoids and then screen them. Still need to scale up, and that's, I think, where this 2D format might come to use. It's probably first going to be pharmacology, so safety, toxicity, transport, blood-brain barrier, gut uptake, these kinds of things. Then new drug development. So by setting up you can actually, if you scale this up, do 100,000 compound screen. So I think that is what the industry will first do. We hope that the diagnostics industry will think about ways of introducing precision medicine type approaches. And it did work very well for Ombion for cystic fibrosis. But it's basically there was only one drug, so it was really important to have something for cystic fibrosis. Second, the patient numbers are so low and they're all treated in centers. So in these centers, you can build up the specific expertise. It doesn't have to be fast, it has to be exact. And that has worked well. But if you think about, 50 new cystic fibrosis patients in Holland, and the 150,000 new cancer patients in Holland every year, it just doesn't work currently in diagnostics. And your question, so explants, fine. If you get enough to do your screen, of course. And it has been tried for several decades already, particularly in hematological malignancies, and it does work well.
Thank you so much, Hans. We have one other question from Shani. I don't know if she wants or he wants to join in and ask, or I can read the question.
Hi, can you hear me?
Yeah.
Yes. Now, I was just wondering, will there be or is there a collaboration between Ombion and Clevers Lab?
Between what?
Ombion
Ombion and
and
Clevers Lab, yeah.
Yes.
I actually gave a lecture for Ombion, so I'm basically an objective supporter because I formerly were not part of Ombion, but we are part of a similar consortium, which I would argue is not as well organized as Ombion does is currently in paper. No.
And Hans, may I suggest, we already said during the cystic fibrosis meeting that we need to talk and hear more about your advices and ideas how to work on all these topics in a coherent way. So I renew my invite to visit Ombion, and we will send an invitation for that. So thank you, because Shani has just joined Ombion. This is why she is asking this question.
Yeah, also I'm a former intern at Clevers Lab, so I worked for Daniel.
Okay.
Yeah, so that's why I was wondering.
Oh, now I see you. Yeah.
Yeah.
Very good. I think we have also another question from Charlie Stevenson, Laila.
Yeah, we do. Charlie, if you want to open your mic and ask your question, or I'm happy to read it otherwise.
Hi. Yeah, it wasn't really a question. It was more just to bring the awareness of the participants on this call to a recently published OECD guidance document on the generation, use, and reporting of research data for regulatory assessments. And I wondered if people were already aware of it or not, because there was a lot of talk about the need to bridge the gap between academia and regulators, and I just thought it could be something interesting, because there's no point on having this great piece of guidance document if the research community don't know about it.
Yeah, maybe Charlie, I come in between. You raise a very important point, because the documents also prepared by the OECD, the FDA, the EMA are very specialistic, and they are also formulated in a very specialistic way. And if this document permeates academia, my a bit blunt answer would be no. And again, here the need to create at least a connection through education and training, and together with the Ministry of Agriculture, we're having a meeting with CBG and EMA, and this need of education is really very much felt also from the EMA and from the CBG. So a very good point that you make, how to build this knowledge and to let this knowledge somehow flow, and to have an intermediary that could also pick up the important concept of this very long, and I dare to say complex, and also document that scientists are not reading every day. So I would like to link your comment to education and training. So we should do this effort. Thank you, Charlie.
Thank you, Daniela. Hans, do you have anything to add to this comment?
No, but I agree. I must say that my scientists probably know very little about how clinical development is done. You know, what an IND is, when you try to register a drug based on phase 3 data, et cetera. So to really understand, and pharmacology in general. So indeed, to have accessible documents on where, you know, what's currently being done, where NAMs could contribute, accessible for scientists, that would be really helpful. We are, I think, in academia very aware there's a lot of movement in the EU, at the EMA, OECD, et cetera. But as mentioned by Daniela, it is a very different language from what we use. And something in between would be really helpful.
Thank you. And that will be maybe the end of our Q&A. I think we don't have other questions. So thank you so much, Hans and Daniela. It was really fascinating discussion. I hope people also enjoyed this. And if you have a final words to share to participants, concluding remarks, do feel free to jump in. And otherwise I will wrap up the session, and people will be able to watch the replay and also send feedback if they have additional question later on. I'm sharing just the screen because we have another session in June coming up, and it will be about NAMS and women health. We are really excited because Dr. Zoher Al-Zadifa from Harvard Medical School will join us to discuss this topic, and we will confirm the date and facilitator soon. If you don't have anything else to add, I'm just really willing to thank everyone, and also say that the replay will be available on our YouTube channel and website, and we will share it as well on LinkedIn. Thank you so much, both of you, and have a great afternoon, and hope that you will have a nice end of the week as well. And with that, we will keep in touch anyways.
Yeah. Thank you for having us, Lilas. Thank you.
Thank you so much. Have a good day.
Thank you. Thank you.